I would like to present to you today the new module of PharmaPendium DDI Risk Calculator. This tool is able to predict drug-drug interactions during the drug development process in order to improve patient safety. During the drug development process, there are different ways of doing predictions of drug-drug interactions depending on where you are in the drug development process, whether that's preclinical or phase 1, 2, or 3 clinical trials. On our side, we use what we call the in vitro, in vivo extrapolations using mainly a mechanistic static model. In this case, this tool is able to predict drug-drug interactions in the clinical trials, in phase 1 mainly, using in vitro data for CYP inhibition, induction, or reaction phenotyping. As well, this tool is able to predict drug-drug interactions when you have clinical data using the same type of approach, this mechanistic static approach. In this case, you could do predictions for phase 1 or phase 2 or 3, or refine your predictions as well as when the drug is already on the market. This tool is developed for pharma industries and for DMPK specialists by DMPK specialists. These types of companies spent 2 years with us to refine the tool and optimize it, as well as to give the best type of prediction that we can do with the mechanistic static approach. So why do we use this type of approach? Because it's one of the ones that is recommended by the FDA according to their guidance from 2020. So it means that this tool is compliant with one of the ways or one of the methods that is provided and recommended by FDA guidance. And as you can see, we're using the same equation that the FDA is using to predict the AUC or area under the curve ratios with this tool. Instead of telling you more about the tool in this way, I'd like to show you exactly how this tool works. Let's use drug-drug interaction within the PharmaPendium suite. By clicking there, I will access directly the drug-drug interaction risk calculator. I will access directly on my own environment where I have the different projects that I'm working on. I can add a new project, for example, that one for today, project 5, and select which type of entry point I want to have them, a victim drug or a perpetrator drug. If your drug is a victim, you can use that one. Otherwise, you can use a perpetrator, inducer, or inhibitor. For today, I will use the victim drugs and enter the name. When you have entered the name, you are able to select the enzymes that are involved in the metabolism of your drug, the 2C9 in my case, and the 2D6. After, you need to enter some data about the fraction metabolized for the 2C9 and 2D6. For example, for my drug, it would be 65% of the total clearance is cleared by the 2C9 in this case, and 25% by the 2D6. When I've entered these values, now I have all the data, so that means there is 90% of the total clearance that is related to the hepatic clearance, and there is 10% of another clearance in this case. What you want to do as well, you can put how you want to retrieve the data for the perpetrator drugs. In this case, this is a repeated dose for the perpetrators with the optimized concentration. And you can select or not the fraction blood and plasma, and the fraction by the microsomes or hepatocyte. It's up to you to decide how you want to do these predictions. From my point of view, I will use the fraction bound because it will give you more predictability at the end for the calculations. So what the tool is doing is going to the library, the large library of drugs. There are 600 or 700 drugs for which we have data to do these predictions, and it is looking into that one to select perpetrators, meaning inhibitors or inducers, and to select as well the right data to do the prediction. It means that you are saving a lot of time in this case because you don't have to enter the perpetrator's data to do the predictions. They are already there, and they are used to do the predictions. This is really key. It's saving time for you to do the predictions, and in this case you can see that in less than a minute you have the predictions already done. The results are the following. There are 247 drugs for which you could do predictions, in this case perpetrator drugs, and they are clustered by therapeutic classes. And as you can see, there are different results. For example, the green bar, meaning there is no risk of drug-drug interaction, so the AUC ratio is close to 1. When you have orange or light yellow bars, they have a bit more of drug-drug interactions. And all these ratios that you are seeing right now are predicted. And you can see that we are providing as well the confidence interval of 90% for the dose of the drugs. Of course, these AUC ratios are related with colors and are coming from the data from the FDA guidance. And you can see that you can what you have on the screen and how the data are clustered and so on. They have a lot of information that you can get from the tool as well. As you can see, there are different, of course, therapeutic classes, and there are drug-drug interactions. If I am interested about this anti-cholesterol drug because it seems to be that there's a high risk of drug-drug interactions for that one, the antidepressant as well, and from others, the antineoplastics are also important. This part of antineoplastic drugs, antioxidant as well as antipsychotic, I can select these therapeutic classes and I go there to see more details of the drug-drug interactions. In this case, you have kind of a forest plot chart where you have the perpetrator drugs and the different doses at which they were given to the people. This is what we have in the database, and as you can see, there are different types of concentrations depending on the drug, and as well, you can manage very easily if there's a drug-drug interaction or not. Again, with the mouse over, you can see that. Also, you can click on these values to see, okay, can I see more details about how the calculation was done? What are the values that were used to do these predictions? As you can see, there are different Ki values that were used for this 2D6 and 2C9. And you also have a link to where the data is coming from, so which articles were used. In this case, if I click on Cmax, I can see, okay, it was coming from this article. This is the condition, and so on. There is no black box in this tool. We are always providing the traceability of the data and the calculation in order to increase the trust for people when they're doing these predictions. So if you go back to this one, there are different filters that can be applied. You can use the therapeutic classes as I did before, but there are ways of refining the result by AUC ratios and also something interesting by probe substrate that are retrieved from the FDA. You could say, okay, give me all the results for 2C9 as a probe substrate, for example, moderate inhibitors, weak inhibitors, and so on. This is exactly what you have at the FDA website, and you have a link for that directly there. And if you select the 2C9, for example, give me everything about 2C9, and you apply it, you'll have a refined list of drugs where you have only the 2C9 pro-substrate inducers or inhibitors. Now, if you want to compare or if you want to assess the drug-drug interaction, you can also compare this one with the actual drug-drug interactions. For example, if I click on the fluvoxamine drugs, there are more details about this drug, but you also have a link to PharmaPendium again to see what the actual what are the actual DDI profiles when the fluvoxamine is used as a perpetrator. By clicking on that, you are going back to PharmaPendium, and you have a list of drug-drug interactions, actual DDIs, where the fluvoxamine compound is involved. Fluvoxamine as a perpetrator there, or on the agomelatine drugs, you have a six-fold increase for those. This is real actual DDI data that is being retrieved from either the regulatory agency's results or from articles. As you can see, there is traceability everywhere. There's a connection with PharmaPendium, and there is always a way to manage the data and also to export the data to Excel if you wish to do that. I will stop there, and we can go back to the slide deck to see how people are using this data and these predictions in real life. If you look at this slide, these are testimonials from different customers, and you can see that, for example, the comprehensiveness of the data increases the confidence of the predictions. This is something that was very interesting. In this case, we get much more reliable results with DDIRC. You get much more information about DDI in a shorter amount of time. This is what they said, and the one that is very interesting as well is that the DDIRC is valuable at the preclinical stage when you don't want to invest a lot in the PK models. Now, while it's great to have this type of feedback, how are customers actually using the tool in real life? And there are a few examples here. One example, one of our customers was using this tool, I would say, to refine the inclusion-exclusion criteria in the phase 2 and phase 3 studies. What does that mean exactly? It means that they have a drug that was in phase 2 and phase 3, and they would like to say, okay, based on the co-medication that we are expecting, what is the risk of drug-drug interactions? Can we include this patient because they have a co-medication, or can we exclude this patient because they have also a co-medication that are at risk for drug-drug interactions? It was used with one of our customers and was also proposed as a strategy to include and exclude patients for clinical trials at the FDA, and it was approved by the FDA, not the tool itself, but the strategy and the prediction that we were providing. Another example here is during a clinical trial submission to a European agency, there was a question from the Swedish agency saying, okay, is the drug that you're developing, does it have potential drug-drug interactions with cytochrome 1A1? At the time, our customer was using DDIRC to quickly assess the predictions for cytochrome 1A1 for their drugs. And again, the mitigation of the risk were approved by the Swedish agency using the predictions provided by this DDIRC tool. And now for the most important example that I think is really key. This is a use case that came from one of our customers recently, and it's really about the DDIRC predictions that were provided to the FDA by one of our customers to support the approval of a new drug and avoid clinical trials. And it meant that the data that was coming from DDIRC was provided to the FDA, and the FDA agreed with the pharma companies to not do some clinical trials. And in this case, this decision was based on the predictions that were provided by this PharmaPendium drug-drug interaction risk calculator. And this is a real key achievement for this static approach, and also for the predictions that are provided in this tool. And so, given these examples, I hope this is something that you will consider trying and discussing with us in more detail. And so with that, I appreciate you listening to me today. Thank you.